Good afternoon to all the esteemed dignitaries, respected speakers, faculty members, research scholars, and participants. On behalf of Mari Lakshman Reddy Institute of Pharmacy, I warmly welcome you all to this inaugural session dedicated to enhancing technology and innovation in pharmacology, and Pharmacy Practice and Association of Pharmaceutical Teachers of India. The 5-day FTP titled Integration of AI and Network Pharmacy in Modern Pharmacological Sciences is thoughtfully designed to bridge the gap between emerging technologies and pharmaceutical education and research. In today's rapidly evolving healthcare landscape, the integration of artificial intelligence, data driven approaches, and network based pharmacy systems is transforming the way we understand drug discovery, patient care, and clinical decision making. By the end of this FTP, I'm sure the participants will carry forward valuable insights such as insights into AI driven advancements in pharmacology, exposure to innovative tools and digital healthcare solutions, understanding of network pharmacy models and their real world applications, opportunities to interact with eminent academics and industries experts. This program aims not only to enhance technical knowledge, but also to encourage research orientation, interdisciplinary collaboration, and academic excellence among faculty members, research scholars, and industry professionals. Before we proceed, I would like to take a moment to be grateful to our honorable convener, Dr. Arunabha Malik, professor and principal. Co-convener, Dr. C.H. Sandip Reddy, HOD, Pharmacology. Coordinator, Dr. S. Balamurli Mohan. Our dear faculty members whose constant support that we had Dr. Suendra
जी हाँ सर
Saha
हाँ
and
सर
Dr.
आप
Saujanya,
पहुंच गए थे
and all the
बट हम
organizing
15 मिनट
members
हो
of
गए
their
वो कनेक्ट
dedicated
करें तो
efforts in
रिजेक्ट
making
करें
this FDP possible.
जी सर
It is now my proud privilege to introduce and invite our esteemed speaker for today's session. I take the pleasure to extend a warm and heartfelt welcome to our esteemed guest speaker, Dr. Saravanan J., Assistant Professor, Department of Pharmacology, JSS College of Pharmacy, Ooty. Dr. Saravanan is a distinguished academician and researcher with remarkable expertise in drug design, network pharmacology, neuropharmacology, and molecular pharmacology. His significant contributions to pharmacological research and education has inspired students and professionals alike. We are truly honored to have you with us today, sir. Your presence and insights will undoubtedly enrich our learning experience and motivate our students to strive for excellence in the field of pharmacy. On behalf of Mari Lakshman Reddy Institute of Pharmacy, I extend a warm welcome to you, sir. We look forward to an enlightening and inspiring session. Thank you. Dear participants, kindly keep your microphones muted during the session. You may post your questions in the chat box and we will take them up during the Q&A session at the end. Thank you so much and over to you.
Am I audible, ma'am?
Yes, sir, you're audible.
Just a minute, ma'am. Is my screen visible, ma'am?
Yes, sir.
Okay. Thank you, ma'am. Thank you very much for your wonderful introduction. And first off, I would like to thank Mari Lakshman Reddy Institute of Pharmacy for having me on this EFDP. And I would also like to extend my thanks to Dr. Arunabha Malik, Principal Sir of Mari Lakshmi Reddy Institute of Pharmacy. I'd also like to extend my thanks to Dr. S. Balamurli Mohan, HOD, Department of Pharmacy Practice, and my sincere thanks to Dr. Sandeep Reddy, sir, HOD, Department of Pharmacology.
Sir,
With this,
excuse me, we can't see your slides, sir.
Oh, okay, sir. Okay, sir. Can you see my slides now, sir?
No, sir. Yes, we are able to see you in person, but we can't see your slides.
Okay, sir. Sir, now, sir?
Yes, sir. Now it is fine. And please put it in slideshow, sir.
Oh, yes, sir. Is it okay now, sir?
Yes, sir.
Okay.
Yes, sir. Perfect, sir.
Okay. Yeah, thank you, sir. So I split this talk on network pharmacology for drug discovery and development into two parts. In part one, I'll discuss the basics of network pharmacology. And in part two, I'll go through general methodology of network pharmacology. Let's get started with basics of network pharmacology. Erum's law is based on the observation that discovery of new drugs is becoming slower and costlier despite improvements in technology like combinatorial chemistry, computer-aided discovery, and high-throughput screening. Normally, when technology improves, the output also improves. For example, if we compare vintage cars with modern cars, there is a lot of difference and improvements. If we compare our telephones with today's mobile phones, there is a lot of difference. The same applies to our laptops and computers as well. So normally when technology improves, the output also improves. But this has not been the case for a while with drug discovery and development. Some of the drugs that were discovered in 18th century and 19th century are still in use. For example, during the COVID times, one of the drugs that we relied most on was paracetamol. We rely still on aspirin, and morphine is one of the gold standard of pain management even these days, but it was discovered in 18th century. Same with digoxin, and there are many other drugs which were discovered in 18th century and 19th century and still are the most important drugs that we use in therapeutics. Technology has improved on site, but the output has not improved in terms of drug design, drug discovery, and development. There are four main reasons for it. The first reason is the cautious regulator problem. No country would want to have a thalidomide-like disaster again under their watch. If something like thalidomide disaster happens again, the government would lose its rule. So every country is making it very tough for scientists to bring a drug into the market. The second reason is inflation. It's a common fact that discovery of new drugs takes over 14 years of time, and more than that, it takes a lot of money. When a pharmaceutical company invests a lot of money and the chances of success is very low, no company would be willing to invest money in drug discovery, and drug discovery is becoming costlier and costlier. And the third reason is a difficulty in discovering drugs that are better than existing drugs. For instance, it would be very difficult for us to discover a statin that is better than atorvastatin. iPhone 16 should definitely be better than iPhone 15. If that is not the case, there is no incentive for innovation. So when we discover and innovate something, it should be definitely better than the existing products. But that is not the case with drug discovery and development. It is very difficult for us to discover a drug better than atorvastatin or Lipitor. But these three reasons are not the most important reasons for a slower trend in drug discovery. The most important reason for this disappointing trend in drug design and drug discovery is the basic research brute force bias. This hypothesis is based on the fact that high affinity binding of a single drug to a single target in a single disease will not offer significant therapeutic benefit. So that is what we rely in classical pharmacology. When we have a drug that selectively binds to a selective target in a disease, it would offer significant therapeutic benefit. But this hypothesis is challenged in network pharmacology. And network pharmacology claims that high affinity binding of a single drug to a single target in a single disease will not offer significant therapeutic benefit. Of course, this approach might offer benefit in simple diseases like sickle cell anemia, Tay-Sachs disease, etc. But this approach has not been offering significant benefit in complex diseases like hypertension, Alzheimer's disease, Parkinson's disease, etc. Complex diseases are those diseases which are caused due to interaction between multiple genes or proteins, and these genes in turn interact with environmental and dietary factors. Human physiology is complex. Human pathology is even more complex. So the idea of magic bullets is great, but in practice, it is probably not going to be the right approach for complex diseases. So when it comes to complex diseases, targeting a single protein will not offer significant therapeutic benefit according to the premise of network pharmacology. So we need to come up with newer trends or newer approach to discover new drugs. One such approach is network pharmacology. Network pharmacology uses two or more drugs that act on multiple targets involved in a disease-causing network of proteins. This definition sounds like combination therapy. In combination therapy, we use a combination of two or more drugs, but network pharmacology is not combination therapy. In combination therapy, we use a group of drugs that act on unrelated targets that control different symptoms. But in network pharmacology, we use a group of ligands that target a group of proteins that are closely interacted within a network. So in combination therapy, we use drugs that target unrelated targets. In network pharmacology, we use a group of ligands that target a group of closely related proteins that play a major role in pathogenesis of that disease. The concept of drugs acting on multiple targets is not entirely new. We have many drugs currently in therapeutics that act on multiple targets to produce their therapeutic benefit. And these drugs that are currently in practice that act on multiple targets are called dirty drugs. Sunitinib is an example of a dirty drug or a drug that acts on multiple targets. Sunitinib is a multiple tyrosine kinase inhibitor, but it also acts on platelet-derived growth factor receptor, vascular endothelial growth factor receptors, CD117 protein, red protein, CD114, and CD135. Clozapine is a D2 receptor antagonist, but it not only acts on D2 receptor as an antagonist, it also modifies the function of serotonin receptors, GABA receptors, muscarinic receptors, adrenergic receptors, and PPAR. Sorafenib is primarily a multiple tyrosine kinase inhibitor, but it also acts on RAS, BRAF, c-KIT, FLT3, vascular endothelial growth factor receptor 2, 3, and platelet-derived growth factor receptor beta. So these are not the only drugs. We have other drugs like tramadol, chlorpromazine, olanzapine, dextromethorphan, kanamycin, axitinib, etc. So these drugs also act on closely related multiple proteins to produce their therapeutic benefit. Okay, before we proceed further, we have to understand how to interpret a network. A network is represented by circles and lines. The circles of a network are called nodes, and the lines of a network are called edges. Every node represents a protein, and every line represents every line is called a edge. So every node represents a protein and every line is called an edge, and it represents an association between two proteins. And the color of the edges represent different things. For example, if a color is rose color, if the line is rose color, it means that the association between two proteins is determined experimentally. On the other side, if it is yellow colored, it means that that association is determined by text mining. So text mining means that if in a paper, if the name of the protein and the name of an other protein, if name of two proteins appear in the same paper, it means that there is an association and it is based on text mining. And the color of the nodes also represents something. So if the nodes are colored, it means that the interaction is first line interaction. For example, these proteins are colored and these are first line of interactors. And if the color of the node is white, it means that they are second level of interactors. If a node is filled with something like this, it means that we know the 3D structure of that protein. If the node is empty like this, we do not know the structure of that protein. We do not know the 3D structure of that protein. So with this, I'll move on to the second part of the talk, the methodology of network pharmacology. So when it comes to network pharmacology, the first step is to choose ligands and a disease that you are going to work on. So the first step is to choose your ligands. The ligands can be natural, synthetic, or semi-synthetic. So it is up to our work. So we can choose a ligand based on aim and objectives of our work. And next we have to choose a disease. So the first step in network pharmacology is to choose ligands and the disease of our interest. The second step is to choose targets based on our ligands. So we have several databases, AI databases, that use the structure of ligands and compare it with existing ligands that bind to a specific target. So it is based on the principle that if two ligands are structurally similar, they bind to the same protein. So based on this principle, we can select the target or predict the targets our ligands can bind with. The third step is to choose the targets that are involved in the pathogenesis of the selected disease. For instance, if we have selected Alzheimer's disease, we have to identify the targets that are involved in the pathogenesis of Alzheimer's. And to do that, we have databases like DisGeneT, GeneCards, and Therapeutic Target Database. So the first step is to choose ligand and disease. The second step is to select targets, proteins, or enzymes that are involved in that can bind to our ligands. And next we have to select proteins or targets that are involved in the pathogenesis of chosen disease. The fourth step is to identify the common targets. So what we do here is we identify those targets that are involved in the pathogenesis of the chosen disease and that can also bind to our ligands. So common targets do two things. They are involved in the pathogenesis of the disease and they can also bind with our phytotargets. So common targets are those targets that have two features. They are involved in pathogenesis of the disease and they also bind to the ligand that we choose. The next step is to find out if there is any relationship or interaction or association between the common targets. To do that, we have to do something called protein-protein interaction network. So the fifth step is to build a network of proteins. And we can do that using a database called Stitch database. And the next step is to perform something called topological analysis. Topological analysis will help us to identify the most important proteins in a network. The next step is to perform gene ontology and pathway analysis. Gene ontology will help us to identify the most important proteins involved in a network and the molecular function, the biological process, and cellular components. So gene ontology will help us to identify the molecular function of the proteins, molecular process of the proteins, and where in a cell they are located. The next one is pathway analysis. pathway analysis will reveal in which pathway our proteins are involved in. So up to step 6 is network pharmacology. So network pharmacology is mostly prediction. So we have to validate our research. So to validate our research, we have to do in silico studies like drug likeness, molecular docking, molecular dynamics, etc., followed by in vitro and in vivo studies. So this is the general outline of network pharmacology. There are 6 steps to perform network pharmacology. Step 7 and step 8 in silico studies, in vitro studies, and in vivo studies are used to validate our results, validate the results we got from network pharmacology. So I'll explain each step in detail. So the first step is selection of ligands. We can work on drug repositioning. If we are working on drug repositioning, our ligand would be an existing drug. If not, we can synthesize ligands. It can be synthetic ligands or semi-synthetic ligands. If not, we can work on plants. So the first step when it comes to choosing the ligands is we have to choose our area. We have to decide whether we are going to work on natural products or drug repurposing or synthetic chemistry. So once you decide it and once you have ligands with you, we can move on to the next step. If you are going to work on drug repurposing or synthetic chemistry, you can skip the next step. If you are working on plants, you have to perform these 2 steps. So we have 3 different options to identify the phytoconstituents present in a plant. The first one is using databases. So there are 2 main databases that can be used to identify the phytoconstituents present in the chosen plant. So the first database is called IMPACT, which stands for Indian Medicinal Plants, Phytochemistry and Therapeutics. So if you go to the home page of this website, you'll see a button called Basic Search. If you click on this button, you'll be taken to this page, and in this page there will be an option to type the name of the plant you have chosen. So it can be any plant. You can type the name of the plant in this box, and if you click search, it will give you a list of phytoconstituents present in this plant. So collect all the phytoconstituents, name of the phytoconstituents, and put it in an Excel sheet and save it for future use. The second option is to use a database called Dr. Duke's database. Go to the home page of this database and you'll find this box, and in this box click on all entities and choose plant. And after choosing plant, type the name of the plant you have chosen and click on search button. Once you click on search button, it will give you a list of phytoconstituents, all the phytoconstituents present in different parts of this plant. So if you are working on a specific part, you can select the specific part only by using this filter and collect the name of all these phytoconstituents and put it in an Excel sheet. And if there are any duplicates, remove it. If you have the chosen plant in Dr. Duke's or IMPACT database, it is fine. But if it is not present in IMPACT or in Dr. Duke's database, you have to move on to the second option. The second option is to do a literature survey using databases like ScienceDirect or PubMed and find out if anyone has reported on phytoconstituents present in the selected plant. If it is present in these databases, if you get the list of phytoconstituents present in your plant from these databases, it is fine. If it is not present, then you don't have any other option. You have to move on to the third option. The third option is to perform the experiments. You have to perform experiments and find out the phytoconstituents present in your plant. So the first step is to identify the phytoconstituents. But if you're working on drug repurposing or synthetic chemistry, you will already have a list of ligands with you. The databases that we use in the further steps will not recognize common names like routine or digoxin or digitoxin, etc. They will recognize a specific format called SMILES format. So you have to convert the common names of your ligands into a format called SMILES format. SMILES format is a chemical notation. It is very similar to IUPAC name. So to convert the phytoconstituent or common name into SMILES format, you can use a database called PubChem. So go to the home page of this website and type in the name. And once you type in the name, you will see a lot of things here. So click on to names and identifiers. And under this, you'll see canonical SMILES. So you have to copy the SMILES format of your phytoconstituent or ligand and put it into the Excel sheet. And if you are going to perform in silico validation, you might also need the 3D structure of this compound. So you can download the 3D structure of your compounds from PubChem itself. So make a Excel sheet of common names and in the next column, put in the SMILES format of the specific compound. If you are working on synthetic chemistry or on drug repurposing, you will only have a small set of ligands. So you can directly move on to the next step. But if you are working on plants, you will get hundreds and hundreds of phytoconstituents, and it would not be practically possible for us to work on all 100 phytoconstituents. So we have to narrow the number of phytoconstituents. So to narrow the number of phytoconstituents, we can perform a pharmacokinetic evaluation study. So to evaluate the pharmacokinetic properties of your phytoconstituents, we can use a database called ADMET 2.0. So go to the home page of this website and click on services. You will see two options, ADMET evaluation and ADMET screening. If you want to perform pharmacokinetic prediction of a single ligand, use ADMET evaluation. If you want to perform pharmacokinetic evaluation on a set of compounds, maybe 100 or 200 or 500 compounds, then you can use ADMET screening. When you click on ADMET screening, it will take you to this box and you can paste these SMILES notations of all your ligands. You can paste a maximum of 500 compounds. You can evaluate the pharmacokinetic properties of 500 compounds at one time. So if you give common names like routine, digoxin or digitoxin in this database, it will not recognize. So you have to convert the common names into SMILES format. So once you have the SMILES format of all your compounds, paste it here and click submit. Once you click submit, it will give you the pharmacokinetic parameters of all compounds in this format. You can click on view button. So if you click on view button, it will give you a lot of parameters. So the first section would be about physico-chemical property. It will calculate the molecular weight, density, number of hydrogen bond acceptors, number of hydrogen bond donors, number of rotatable bonds, ring structures, etc. And it will also give a lot of results about medicinal chemistry like Lipinski rule, Pfizer rule, GSK rule, ease of synthesis, etc. And it will also give you results about absorption, distribution, metabolism, excretion, etc. So if the results are in green, it means that the value is acceptable. If the results are in red, it means that the values are very poor and not acceptable. If it is in yellow, it is in mid value. So if you click on this eye icon, information icon, it will give you a brief explanation of what that particular parameter means. For example, if you click on this icon, it will show that it measures the absorption through Keker-2 barrier. And it will also say whether what the volume of distribution is, what the blood-brain penetration is, and it will also calculate the clearance value and half-life value, etc. So based on pharmacokinetic evaluation, we can choose a ligand for further studies. If not, we can reject it. For instance, if you are working on Alzheimer's disease and if a drug does not cross the blood-brain barrier, there is no point in working on that ligand. So you can reject all the ligands that fail to cross the blood-brain barrier. So by performing pharmacokinetic evaluation, we can narrow down the number of ligands. So now you might have hundreds of ligands or 40 to 50 ligands only if you are working on plants. But if you are working on drug repositioning or synthetic chemistry, you will might have only a handful of drugs. So now we have to identify those proteins that our ligands can bind to. To do that, we have three main databases: Swiss Target Prediction, Stitch Database, and Binding DB. So these databases will predict what proteins our ligands can bind to. These databases work on the principle that drugs that are structurally similar will bind to similar proteins. So the first database that we use for this case is Swiss Target Prediction. So go to the home page of Swiss Target Prediction and you'll see a box to paste your SMILES notation. So copy the SMILES notation of your ligand and paste it here and click on Predict Targets. So once you click Predict Targets, it will give you a list of proteins like transporters, channels, enzymes, or transporters that our ligands can bind to. So copy all these proteins and put it in an Excel sheet. The second database that is used for prediction of phytotargets is Stitch database. So go to the home page of Stitch database and type Mangiferin here and select organism as Homo sapiens and click on search button. So once you click on search button, this database will also give you a list of proteins that our ligand can bind to. So again, copy all these protein name and put it in an Excel sheet. The third database is called Binding DB database. So go to the home page of Binding DB database and click on Special Tools. So after clicking on Special Tools, so click on Find My Compounds Targets. So once you click on Find My Compounds Targets, it will take you to this page. And in this page, you will see a box to place your SMILES notation. So place the SMILES notation in this box and click on OK. And then click Go. So this database will also give you a list of proteins that our ligands can bind to. So at the end of this step, you will have hundreds or thousands of targets that our ligands can bind to. So all these targets are called phytotargets. So the next step is to predict the disease targets. Disease targets are those proteins that play a major role in the pathogenesis of a disease. So if you select hypertension, if you have selected hypertension, these databases will give you a list of proteins that are involved in the pathogenesis of hypertension. If you have chosen Alzheimer's disease, it will give you a list of proteins that are involved in the pathogenesis of Alzheimer's. So the three main databases that are used in predicting disease targets are DisGeneT, GeneCards, and Therapeutic Target Database. So first database is DisGeneT database. After visiting the home page of DisGeneT database, you'll see a search button. So click on this search button. It will show you a box and type the name of disease you have chosen. It could be hypertension or Parkinson's disease or Alzheimer's disease, whatever it might be. So once you click on search, it will give you a summary of gene disease associations. If not, it will ask you for, there are several options. The first one is relevant in network pharmacology. So select the first option, summary of gene disease associations. So if you click on summary of gene disease association, this database will give you a list of proteins that are involved in the pathogenesis of Alzheimer's disease. If you have typed in hypertension or angina pectoris or myocardial infarction, this database will give you the list of all proteins that are involved in the pathogenesis of the chosen disease. For instance, I have put in Alzheimer's disease and it gave me a list of 3,397 proteins that are involved in the pathogenesis of Alzheimer's disease. So if you look at this list, the first protein is amyloid beta. So it's a well-known fact that amyloid beta is involved in the pathogenesis of Alzheimer's disease. The same with apolipoprotein E. Apolipoprotein E is a major risk factor in Alzheimer's disease. So the second database that is used for predicting the disease target is GeneCards. So go to the home page of this database and click on Malacards. So once you click on Malacards, it will take you to the next page. And at the bottom of this page, you see Explore a Malady. Under this, you see an empty box. So type in the name of disease you are working on and click on Go. So once you click on Go, so this database will also give you a list of proteins that are involved in the pathogenesis of a disease. So copy down the name of all the proteins and save it in an Excel sheet. The third database that is used in the prediction of disease targets is therapeutic target database. So go to the home page of therapeutic target database and type in Alzheimer's disease or any disease you have chosen and click on search. So this database will also give you a list of all proteins that are involved in the pathogenesis of Alzheimer's disease. So this database lists out M1 receptors or acetylcholine receptors, and it is a well-known fact that acetylcholine receptors also play a major role in Alzheimer's disease. Okay. So after this step, we have a list of phytotargets and a list of disease targets. So all these targets
One
individually.
thing, Doctor,
Yes, sir. Hello.
Dr.
Sir.
Saravanan, one question to you.
Yes, sir.
How to download the whole data of disease target? Suppose
Sir,
if I work on Alzheimer disease,
yes, sir. Yes, I got it,
just
sir.
like
Sir, in therapeutic target database, you can download it. In Malacards, you can download, but in DisGeneT, we do not have an option to download the resource. We have to do it manually.
okay, thank you.
Yes, sir. So in this case, we have to do it manually, whereas in this database, you can see a download icon. If you click on that download icon, it will download the entire resource in Excel format. So if the download option is not available, we have to do it manually. We don't have an option, sir. So after completing this step, the third step, we'll have a list of phytotargets and a list of disease targets. The next step is to identify the common targets. Common targets are those targets which are involved in the pathogenesis of the disease, and they can also bind with our ligands. For example, in this case, the number of targets for our ligands is 468 and number of disease targets is 2033, and the common targets are 44. So what this 44 means is that these 44 proteins are involved both in the pathogenesis of the disease and they can also bind with our ligands. So this 40, the remaining 2033, sorry, 468 minus 44, the remaining 424 proteins cannot bind with our ligands. So only these 44 proteins are capable of doing two things. So if you block these proteins, if you modify the function of these 44 proteins, it will have a beneficial effect in the disease. So to identify the common targets, we use a database called Venni database. So go to the home page of this database and you'll see this page. So the first one would be list 1 and second one is list 2. So you can rename it. You can delete list 1 and type phytotargets. You can delete list 2 and type disease targets. So once you complete it, if you click on enter, it will find out the common targets. And if you click on 44 in the results section, it will give you the list of common 44 proteins. So the next step is to find out, is there any relationship or association between these 44 proteins? If there is no relationship between these 44 proteins, there is no point in working further. So our next aim is to find out if there are any relationships between these 44 proteins. To do that, we use a database called string database. So go to the home page of string database and it would look like this, and click on multiple proteins. So copy the name of all those 44 proteins and paste it here. And you can select the organisms as Homo sapiens. If not, the website itself will detect it as Homo sapiens, and click on search. So if you click on search, it will build a network called protein-protein interaction network. So what this network explains is, it explains the relationship or association between the 44 proteins. So if two proteins are connected to each other by edges, it means that those two proteins have a relationship between them. So these circles are called nodes and these lines are called edges. The circles represent protein and the edges represents the association. The color of the edges mean different thing. For instance, if the color is rose colored, it means that this relationship between these two proteins has been determined through experiments. So someone has performed an experiment and established a relationship between these two proteins. If it is in yellow color, it means that the relationship is through text mining, which means that if the name of these two proteins CA12 and CA4 appear in the same published paper, it means that there is some association. Next, there is a green color, which means that they are very close to each other in terms of genes. So on and so forth. So if you look at these two proteins, they are not associated with any of the remaining 42 proteins. So we have one protein here, which is also not associated. So we have to work on string network to build, string database to identify the protein-protein interaction network. So once you build protein-protein interaction network, still we have a large number of proteins. For example, in the previous network, we have around 40 proteins. It is practically not possible for us to work on 40 different proteins. It is impossible to find a ligand that binds to all of these 40 proteins. So it is not possible. So the next step is to identify the most important proteins in this network. To identify the key proteins in a network, we have to perform something called topological analysis. Topological analysis is the examination and interpretation of relationship within a network of proteins. What topological analysis does is, it helps us to identify the most important proteins in this network. By identifying and modifying the function of most important proteins in a network, we can modify the functions of this entire network. So topological analysis helps us to identify the key proteins. So we measure three important parameters while doing topological analysis. They are degree centrality, closeness centrality, and betweenness centrality. So degree centrality is the measure that counts the number of connections a protein has with other proteins. For example, if you look at this network, there are 7 proteins, but the protein in the middle, the red color, has a degree of 6, which means that this red color protein is connected to 6 other proteins in this network. whereas this protein is connected only to one protein. So the degree of this red colored protein is 6, whereas the degree of all other gray colored proteins is 1. So whenever we want to choose the most important protein in a network, you can select a protein that has the highest degree value. So degree centrality is based on the number of connections a node has with other nodes in the network. The next one is closeness centrality. Closeness centrality measures the average length from one node to all other nodes in the network. So basically it measures which protein is located very close to all other proteins in a network. So if you compare this protein with this protein, they are very far from each other. If you compare the distance between this protein and this protein also, they are very far from each other. But if you take these two red colored proteins into consideration, they are very close to all other proteins in the network. The next one is betweenness centrality. Betweenness centrality identifies the critical points of control or communication. It identifies the protein that controls the communication, the most important point of control in a network. So if you look at this network, so we have a protein here and a protein here and a protein here. So if you block this protein, it will not have much impact on the remaining proteins. But if you block this red colored protein, it will have an impact on all other proteins in a network. So degree measures the number of connections a node has with other nodes. Closeness centrality identifies the protein that is located very close to all other proteins in a network. Betweenness centrality identifies the most important control point, the protein that controls a network. So to perform topological analysis, we need a software called Cytoscape. It is a free software. We can download it and you can install it. So you have to go back to string network again, and you under the network, you'll see a tab called export. So click on export. Once you click on export, a tab will appear. Send network to Cytoscape. So click on send network to Cytoscape. So once you click on this tab, it will take some time, maybe 5 to 10 seconds. And after that, this network will appear in Cytoscape software. So once you have the network in Cytoscape software, you have to click on tools. So when you click on tools, you'll see this tab, and under that you'll see analyze network. When you click on analyze network, the software will analyze the network. It will perform topological analysis. It will calculate the degree. It will calculate the closeness centrality and betweenness centrality of this network. And it will help us to identify the most important proteins in a network. So once you click analyze, it will give you a summary of results. So the number of nodes or number of proteins in this network is 110. The number of edges is 196. The average number of neighbors is 4.267. Network diameter is 8, etc. So all these parameters are not very relevant to us. So what we need is degree centrality and betweenness centrality and closeness centrality. At the bottom of this page, if you drag this tab up, you'll get closeness centrality, betweenness centrality, and degree centrality. So you can use any one parameter or two parameters. It would be very difficult for us to use all three parameters because one protein might have a high degree value, but it might have only a very low betweenness value. So you can take any one centrality, any one topological measure into consideration, and you can select top 10 or top 5 proteins for further studies. The next step is to perform gene ontology. Gene ontology is a study that associates proteins or genes with their molecular function, biological process, and cellular component. When we perform gene ontology, we can say whether our proteins are really relevant to our chosen disease. For instance, if you have COX-1 gene in your list, and if you see the function of COX-1 gene is mediation of pain, and if you are working on analgesics, it makes sense. So gene ontology will tell you whether your proteins are related biologically in molecular or in cellular way. For instance, if you're working on Alzheimer's disease and if all of your proteins are present in only muscle cells, it does not make any sense. On the other side, if the chosen proteins are present in neurons, it definitely makes sense. So by performing gene ontology studies, we can identify the molecular functions, biological process, and cellular components, and it will help us to prioritize potential drug targets for further experimental validation. So to do that, we have a database called Shiny Go database. So go to the home page of this database. So at the end of topological analysis, you will have only a minimum number of proteins. So if you have around 10 proteins, paste the name of all the 10 proteins here and click on submit. So once you click on submit, it will give you the results. So the proteins that I selected are involved in response to ethanol, response to lipopolysaccharide, negative regulation of secretion by cell, response to organic cyclic compound, etc. So these are the biological processes that my proteins are involved in. Next, moving on to molecular function, the proteins that I selected are involved in histone deacetylase activity, NAD dependent histone deacetylase activity, nuclear receptor activity, DNA binding, transcription factor binding, etc. So if you look at all these proteins like STAT protein, heme oxygenase, myeloperoxidase, androgen receptor, etc. These proteins are mainly involved in pathogenesis of cancer. So you can see all these functions are also related to cancer. Next, if you click on cellular component, it will tell where in the cell these proteins are located. It will tell whether these proteins are located in chromosomes, whether they are located in chromatin, whether they are part of histone deacetylase complex, etc. It will also tell whether whether it is located in the nucleus, whether it is located in the ribosomes or mitochondria, etc. So as I said earlier, if you're working on Alzheimer's disease and if you find out that your proteins are present in cell body, axon, dendrites, etc., it makes a lot of sense and it tells you that you are going in the right direction. If the cellular components are not relevant, then it does not make much sense. The next step is to perform pathway analysis. So pathway analysis helps us to identify the pathway in which our proteins are involved in. For example, if you are working on pain, if you are working on analgesic activity, and if you find out that your proteins, your common targets are involved in arachidonic acid pathway, platelet aggregation pathway, it becomes very relevant. So to do that, we use a database called Reactome database. So go to the home page of this database and paste the name of all the proteins. After that, click on Go. So paste the name of all the proteins and again click Continue. And here you'll see an Analyze button. Click on Analyze. So now it will give you a list of molecular pathways, pathological or physiological pathways our proteins are involved in. So this is the name of all the proteins. So if you have pasted a name of 10 proteins, here if you see 4, it means that the 4 out of 10 proteins that you pasted in the previous step are involved in this pathway. So 4 out of 10 proteins are involved in SOMylation. 2 out of 10 proteins are involved in regulation of heme oxygenase. So in this pathway, there is a total of 184 proteins, 193 proteins. So out of 184 proteins, the 4 proteins in our list are involved. It will also give you a summary of pathway, and the colors that are highlighted in green are the proteins that we have pasted. So if we do a pathway enrichment analysis, we can find out the exact role of the proteins in a pathway or pathogenesis of a disease. So network pharmacology is all about prediction. So we have to validate our results. So whatever we did so far is mostly prediction. So predictions are not conclusive. So we have to validate our results through in silico studies, in vitro studies, and in vivo studies. So we can do in silico studies like molecular docking, molecular dynamics, MMGB assay, etc. And we can do cell line studies in vitro, and we can also do animal studies. So if we complete all these studies, it will complete our network pharmacology analysis. So network pharmacology is not foolproof. It is not perfect. We have a lot of limitations in network pharmacology. The first limitation is data integration and quality. For most of our research in network pharmacology, we rely on databases, and these databases get data from published papers. We do not know the quality of published data. All published data is not of high quality, and we could not say that there is honesty in those data. The first problem is data integration and quality. The second one is biological complexity. So even though network pharmacology gives much importance to the complex nature of our human body, we cannot still capture it. So biological complexity is also a problem. The next one is predictive accuracy. So all the results that we got from network pharmacology are predictions, and we are not sure whether whether the predicted results are 100% accurate or not. If the results are 100% accurate, then it is fine. But unfortunately, we do not know how accurate the results are. The last one is lack of standardization. So all those predictions are not standardized. All those things are just predictions. So I'll go through the methods once again. So the first step is to select ligands. So you can work on synthetic chemistry, semi-synthetic chemistry, or natural. If not, you can also work on drug repurposing. The second step is to identify the ligand targets. We can identify the ligand targets by using databases. The next one is to identify disease targets by using databases like Dysgene, Therapeutic Target Database, and Malakars. The next step is to identify the common targets. We can identify the common targets using a database called Venni database. If you paste the list of phytotargets and common targets and click on OK, it will give you a list of common targets. After identifying the common targets, you have to build protein-protein interaction network. We can use string database to build protein-protein interaction network. After building protein-protein interaction network, we have to identify the most important proteins in a network. To identify the most important proteins in a network, we have to perform topological analysis. We have to identify the degree centrality, betweenness centrality, and closeness centrality. To do that, we use a software called Cytoscape. It is a free software. You can download it and you can use it. The next step is to perform gene ontology studies. Gene ontology studies will help us to identify the biological process, molecular function, and cellular component of our common proteins. The next step is to perform pathway analysis. Pathway analysis will help us to identify the pathways, the biological pathways or disease pathways our proteins are involved in. So after completing pathway analysis, we can move on to in silico studies like ADMET evaluation, drug likeliness, molecular docking, molecular dynamics, etc. And finally, we can move on to biological validation, in which we can perform in vitro and animal studies. So to conclude, network pharmacology does not accept the fact that high affinity binding of a single ligand to a single receptor will produce significant benefit. In most complex diseases, a single protein is not the cause of a disease, rather, it is a network of proteins. So, network pharmacology concludes that if we modify the functions of two or more closely related proteins in a network, we can reap more therapeutic benefit and cure a disease more effectively. So these are some of our publications from network pharmacology area. So that's all for this presentation. So if there are any questions, I'm willing to answer.
Uh, Doctor,
Yes, sir.
can you please describe how we can use these outcomes of data in our research article?
Sorry, sir, I didn't get you. Can you come again?
Uh, Actually, we are trying to do some research on
Okay,
using
sir.
phytochemicals and disease target.
Okay, sir.
Then,
Okay.
how we can use these data in our research article?
Which data, sir?
Uh, Data of protein-protein interaction
You
or
can
pathways.
you can download the image. You can download the protein-protein interaction network from string database itself, and you can
Okay.
give that image itself. So if you look at this string database,
Yes.
if you look at this
Okay.
network, so you'll see download option. So if you click on settings, you'll see a download option. You can download this image in any format. You can download in JPEG format or in any format you want. You can also change the color. You can do a lot of modifications.
Okay. Okay. Uh,
And
uh,
yes, sir.
Today is a time constraint, but I
Okay.
hope to join with you in personal so that I
Yes, sir.
can
Sure.
learn more things
Yes,
from
sir.
you.
Yes, sir,
Okay.
sure. I also shared a Word document. If you look at that Word document, you'll find the link for all the databases and a step-by-step procedure. So if you follow
Yes,
that
sir.
procedure, you'll get all the data. You can also get
Yes,
in
sir.
contact with me, sir. No problem, no issues.
Okay. I understand there's all things, but
Oh,
if I feel any difficulty during my
okay.
study, I'll surely
Okay.
contact you.
Yes, sir. Sure, sir. Sure.
Thank you.
Sir, another question from another participant. How
Yes,
can we
ma'am.
determine which ligand interacts most effectively with a target protein when multiple ligands are analyzed?
We cannot do that in network pharmacology itself. We have to move on to in silico studies. We have to perform molecular docking. So if we perform molecular docking, we'll get an idea whether our ligand binds with our protein or not. And in the next step, we have to do molecular dynamics. So molecular dynamics will help us to find out whether our drug receptor complex is stable or not. So by performing in silico studies like molecular docking, molecular dynamics, MMGB assay, etc., we can find out which ligand binds more effectively and stable and makes a stable bond with our protein.
Okay, sir. Moving on to the
Uh,
next question. In
Yes,
topological
ma'am.
analysis, which
Okay.
parameters are most reliable or appropriate to consider?
Ma'am, different authors rely on different. There is no set rules for it. We can rely on degrees. Most published papers, the authors rely more on degree. So degree protein which has a highest degree value interacts with many proteins. So most authors rely on degree, but it is not wrong to rely on closeness centrality or betweenness centrality either. So there are no set rules. But if you want to be on a safer side, it is degree.
Okay, so another question. Is it possible to identify which biological process or disease
Yes, ma'am.
pathways are mediated by specific proteins among the top 10
Yes, ma'am.
identified molecules?
Yes. After performing topological analysis, you have to restrict the number of proteins to 10. not more than 10. So once you have 10 proteins, if you perform gene ontology studies, it will give you a list of molecular functions of those proteins, biological processes, and cellular components. For example, if someone is working on Parkinson's disease, we expect those proteins to be present in neurons. So if you perform gene ontology studies and if you click on cellular component, it will list out where your proteins are present inside a cell. So if you see a lot of neurons there, like in axon, dendrites, or cell body, etc., it means that our results are relevant. And if you look at molecular function and if they are involved in metabolism of dopamine, then it becomes relevant.
All these
Uh,
three are the queries
for
from Dr. Akiraju Sudhir. Hope the queries
Yes,
are
ma'am.
cleared. So
Okay,
next
ma'am.
question is from Pradipti.
Okay.
From a pharmaceutics perspective, when a network
Okay.
pharmacology identifies multiple active compounds in a polyherbal system, how do we
Okay.
address the formulation challenge of combining molecules with drastically different physiochemical properties like solubility, stability, and permeability into a single dosage form?
That lies outside the prospect of network pharmacology, ma'am. Network pharmacology is not about formulation.
Okay, sir.
So network pharmacology helps us to identify a group of 2 or 3 proteins that are closely involved in the pathogenesis of a disease. So it has not got nothing to do with formulation, ma'am.
I think she's having the another question. How do you
Yes, ma'am.
guide
Sure.
students in deciding the appropriate cutoff values for parameters like degree centrality, betweenness, and confidence scores during target and
Yes, ma'am.
interaction filtering?
We cannot.
Given that
We
there
do
is
not have a.
no,
Yes, ma'am.
yes,
Yes, ma'am.
please continue.
We do not have a set values like degree should be greater than 10 or degree should be less than 5 like that. So in a given network, we try to identify the most important proteins. So if only one protein has interaction with 2 other proteins, 2 itself is a high number. But if your network is too large, you will
Ending.
have a protein that interacts with 5 or 10 other proteins. So there are no set values. We try to identify the protein with highest degree value.
Other last
It can
question.
be 6 or it can be 60 or 600. Yes, ma'am.
Thank you, sir. That is the last
Okay.
question.
Yes, ma'am, sure.
So, any more queries? Dear participants, please feel free to ask the questions.
Sir, can we directly get a help of AI tools like Gemini to download the common targets of that particular disease rather than going on the database?
Any database is exclusively designed for this purpose. I think you can also get it from AI tools, but here you'll get a good image and other things. And in published works, you'll always see Venni being used for this. Your voice is not clear, sir.
Thank you very much, sir, for
Okay.
that truly insightful and enriching session. Your
Thank you,
deliberation on
ma'am.
the integration of emerging technologies like artificial intelligence with interdisciplinary perspectives have given us a deeper understanding of how innovation is shaping the future of pharmacological sciences. The way you connect your vast experience with the evolving concepts of network driven and technology enabled approaches has been both inspiring and thought provoking for all of us. I'm sure our participants have gained meaningful insights that will greatly contribute to the academic and research pursuit. We will now end the session. Participants, thank you so much for your questions, queries, and hope you all got your answers. Thank you, sir, for patiently answering all the
Thank you, ma'am.
queries and for sharing your
Thank you, ma'am.
valuable knowledge with us.
Yes, ma'am. Thank you.
Thank
Thank
you.
you very much.
Dear participants, feedback link will be posted in the chat box. Please have a look on it.